students, we are going to get back to the nephron. So we are about halfway through, and we just have to finish up with the distal convoluted tubule and the collecting duct today. Before we do that, let us do recap a little bit. Let's start with this figure over here, and let's just kind of remind ourselves what's going on within this figure. So this is our functional diagram of a nephron all stretched out just so that we can see what's going on. Anything that is marked in blue is going to indicate that that is a substance that is reabsorbed. So again, remember, we already absorbed the good stuff in our digestive system. We filtered it back out in the blood, and then we need to reabsorb the good stuff again. So 65% of the filtrate, so water, salts, organic molecules, glucose, amino acids, things like that, those absolutely need to get reabsorbed, and they do in abundance in the proximal convoluted tubule. Anything that is marked in green on this figure is showing secretion. So these were either things that were too big at the filtration membrane, like any sort of drugs. Maybe you were taking penicillin from an infection, or maybe you are on opioids for pain management. So you've got to get your morphine back in there and get rid of it. So some things are maybe just too big, or as we find out when we get to the distal convoluted tubule, it's just going to depend on what our body needs at the moment. Is sodium too low? Is blood pressure too low? Is potassium too high? So those are reasons we might secrete things after we get to the end and realize we need to make a little adjustment here. Okay, so again, just a little quick recap. So proximal convoluted tubule, really concerned with reabsorbing as much of the good stuff as possible. Our friend the nephron loop here, or loop of Henle, this thing is very specific. So in the descending segment, or the thin segment, as we're coming down into that renal medulla and exposing ourselves to that renal medullary osmotic gradient, we are only reabsorbing water on the descending thin side. On the ascending side, in that thick segment, that is where we are primarily reabsorbing sodium and chloride. And again, it is that coordinated reabsorption of water and then sodium and chloride in those regions that is going to create and preserve that medullary osmotic gradient. I want to review that, and I also want to make sure it makes sense to us, because we're going to do the DCT, but looky here at the collecting duct. The collecting duct is going to flow down into the renal pyramid, and it's going to be exposed to that same gradient that the nephron loop is. So it does us a good job here just to make sure that we understood the things that we covered in the last lecture, so that we understand what the collecting duct is up against here. So again, just to refresh, medullary osmotic gradient. As we're coming from the PCT into the nephron loop, right here, as we are doing that, we are crossing into the renal medulla, and the osmotic concentration changes here. So coming into the renal cortex, the relative concentration of blood, and then also just the interstitial fluids in the tissues, that's going to be about 300 milliosmoles. And that's really what it is pretty much in your whole body. That's where you want to be. That's what you're striving for your normal. As we take a trip down into the medulla and travel through a renal pyramid, as we get closer and closer to the tip, that renal papilla, the concentration goes up, and we end up with 1200 milliosmoles at the deepest part of the renal medulla. Remember, that's saltier than a mouthful of seawater. So in really strategically pumping out water, right, as we head down in the nephron loop, and really strategically pumping out sodium and chloride as we head up top, this is really going to create and preserve this gradient. So we want to maintain this, okay? So we really set this up with the countercurrent multipliers. So that's basically what's going on in the nephron loop. and we're able to preserve it with the vasa recta because, you know, as this blood is coming in and we're trying to reabsorb, that water and salt has to go somewhere. And we end up with really, really salty, you know, saltier than seawater blood at the bottom of that hairpin turn. But because water and solutes are able to kind of juggle in and out as we are moving through the renal medulla, we end up preserving that gradient. So the blood that has reabsorbed all those water and solutes, it comes out just about even, pretty close to 300 milliosmoles. So they're preserving that gradient and they're acting as countercurrent exchangers. And we want to preserve that gradient because, again, we're going to focus on the collecting duct in the very near future. So we want to take advantage of that gradient. And if we need to reabsorb a little bit more water or salt, we're going to exploit that. Okay, so we are going to move on to the collecting duct here and talk about how that's going to be utilized. Okay, again, go back and watch that video if you need a little bit more. So for where we are now, okay, let us go ahead and start fresh with the distal convoluted tubule, and then I promise we will get to the collecting duct. So the distal convoluted tubule is circled here in red. And at this point, now that we've already done a lot of reabsorption in proximal convoluted tubule and adjusted our water and salt content in the nephron loop, we only have about 15 to 20% of the filtrate volume left. So we're doing really good. However, you still don't want to be peeing out 15 to 20% of your filtrate volume. We want to get down to 1 to 2 liters. We would be in the bathroom all day if we were still peeing out this volume. So it's really critical that we reabsorb this last little bit of water, this last little bit of sodium, and then we make some other adjustments here for things like maybe drugs or vitamins that we took, or perhaps we have an issue with our potassium or our hydrogen balance. Okay. So we've started with something that looked a lot like blood, 300 milliosmoles, really just blood minus the cells and the proteins. And now we have something that's a lot more urine-like by the time we get to the distal convoluted tubule. Again, even though the blood that was being taken away with the vasa recta and the peritubular capillaries, that was about 300 milliosmoles. The concentration here, as we go into the DCT, right, we're like at about 100 or 150 milliosmoles, if you were concerned about that. So we're actually a little bit hypoosmotic here. So we're in a position if we needed to secrete things in there, or if we need to reabsorb a little bit more water, we can certainly do that. Urea, which is a nitrogen product, and nitrogen wastes are now our main solutes. And if you remember what the goal was, okay, if you remember what the goal was here, it was really to strategically get rid of a lot of those nitrogen wastes because we just can't do it with the equipment that we have in the digestive system. So DCT has a couple of nicknames. Sometimes we call it the secretion segment because this is where a lot of secretion is happening. So again, draw your attention to the green box here. And again, things that were too big to find their way out, like drugs, they're going to be secreted in here. And then what we'll spend just a couple minutes with these mechanisms, potassium, hydrogen, ammonium ions. Again, ammonium, that's going to be something that contains nitrogen, so it fits the category of a nitrogen waste. And then we have potassium. So we know potassium is really critical for maintaining our salt and fluid balance in the body. If we didn't have the right concentration of potassium, we couldn't fire an action potential and we would be dead. So the DCT is really aimed at making sure we don't end up hyperkalemic. Hyperkalemia is whenever we have too much K, potassium, in our body. So this is a place where we can secrete extra K. We know that the lung was heavily involved in acid and base balance because every time we accumulate CO2, we're actually going to shift that CO2 into hydrogen and a bicarbonate ion. So this is another place, if we're not breathing fast enough to get rid of our CO2, we can actually secrete a little bit of hydrogen into the distal convoluted tubule and will help remove that excess hydrogen, thereby dropping our CO2 concentration and helping the lung out whenever we get in a situation where the lung cannot keep up with that acid-base balance. We're going to use the carbonic anhydrase reaction here. So again, don't forget about that thing. The other nickname for the DCT is the hormone sensitive segment. So we've already heard about aldosterone. We're going to walk through part of that mechanism one more time. Remember, aldosterone is going to be kind of our salt saving hormone. So this is going to promote additional sodium reabsorption. And we'll also talk about the other half of that mechanism that could help out with potassium secretion. Okay, so we'll look at that. And then antidiuretic hormone is a big one. This is going to help with additional water reabsorption, not just in the DCT, but this is when we're going to head to the collecting duct, and that's where that osmotic gradient is going to come into play and help make this thing work. There are two other hormones that work here as well. I've kind of grayed them out to kind of downplay their importance. Not that they're not important, but they're not a big focus right now, or we've discussed them elsewhere. So atrial natriuretic peptide, again, anything you see natriuretic, look Na, that's sodium hiding in there. So atrial natriuretic peptide is going to do the opposite of aldosterone and actually promote sodium loss. You would only want this if your blood pressure was really high and then you needed to pee out some extra sodium and hopefully pee out some extra water, because water follows salt, and that would reduce your blood volume and help bring your blood pressure down. Okay, obviously acting in the heart, right, atrial. Parathyroid is grayed out, again, not because it's not important, but we've already talked about this either in the endocrine system or with bone, because parathyroid is going to act with calcium reabsorption. So if we got in a situation where we didn't quite have enough calcium to maintain muscle contraction or fire action potentials or signal, because calcium does lots and lots of signaling, we would reabsorb here, because we'd rather reabsorb it from our tubular fluid than to have to steal it from our bones, which we absolutely would take calcium for our bones if we had to. Parathyroid hormone dictates that. Okay, so let's talk about aldosterone first. So these major consequences of aldosterone, and again, this is going to be twofold. We are either going to try to increase sodium reabsorption, or we're going to try to increase potassium secretion and getting rid of it. So it just depends on what the needs of our body are. The reason these two things are linked shouldn't be shocking. They are linked by the sodium potassium ATPase, right? One of our favorite ion exchangers, and also something that is active transport because we see ATP in the name. So this mechanism is located in the principal cells of the DCT. Go back and review your cell types if you're a little confused about what the principal cells were, but they are a main component of the distal convoluted tubule, one of two cell types. And again, on a daily basis, your body is probably more concerned with just reabsorbing enough sodium. Again, water follows salt, so if we gotta stay hydrated, gotta reabsorb that last little bit of sodium so we can help reabsorb that last little bit of water. So in a normal daily basis, that's probably what's happening to you. Again, we have to signal to the adrenal cortex, so the outer part of the adrenal gland, and specifically, again, we had those three layers of tissue. So we're going for the mineralocorticoids, aldosterone, getting released from the zona glomerulosa. Now that makes sense that we call it the zona glomerulosa, because we've talked about the glomerulus so much. That should tie that to the kidney for you. Okay, so then we get aldosterone released from the zona glomerulosa layer in our adrenal cortex. The target is going to be the kidney tubules, right, distal convoluted tubule, and that is really going to lead to more sodium reabsorption. That's going to get us back up to homeostatic levels, and again, probably help us reabsorb a little bit more water. Other side of the coin, again, we cannot have too much potassium in our body. If we have too much potassium in our body, that will hyperpolarize our cells. That will make it harder to fire an action potential. If we cannot fire action potentials, we will die. So there is a reason why potassium is a #1 main ingredient of lethal injection. So if we were trying to put someone to sleep or kill them with a lethal injection, we would absolutely include a very high concentration of potassium in that cocktail. And that is why our kidneys, just on a daily basis, if we end up a little bit hypokalemic, right? Again, hyperkalemic, look for the K, look for the K. This has to do with potassium. What this would do would then be to help rid the body of excess potassium. So again, that target would again be the kidney tubules, and because the aldosterone is stimulating this mechanism here in the principal cells by promoting sodium-potassium exchange, every time we reabsorb sodium molecules, we're going to excrete potassium and get it into the urine and get rid of it. And that's going to restore homeostasis. So hopefully we're back to a nice safe level of potassium and we don't hyperpolarize our cells and die. Okay, don't panic. This is a busy figure. There are just a couple tiny key takeaways that I want you to get from it. So we need to talk about hydrogen ion removal and conservation. Basically, this is described as acid-base balance. Okay, so we've already talked about acid-base balance in terms of the respiratory system. We know, right, that CO2 is tied to hydrogen ion concentration via the carbonic acid equation. So if we have too much CO2, a waste product in our body, we're going to end up with too much hydrogen ion. The easiest thing we can do is just to breathe more, to get more CO2 out of our body, and that will help bring our pH back up to normal and reduce the hydrogen ion concentration. This mechanism in the kidney, this can also help us out. So if we are just breathing as much as we can and we're not able to correct the problem, or maybe we've got bigger problems, maybe we're a diabetic and we're in a ketoacidosis or some type of metabolic acidosis. So we've got problems here. For some reason, the acid concentration is going up in our blood. And maybe it's not just CO2's fault. Maybe it's because we have, you know, an underlying medical condition that is knocking us out of a homeostatic balance. Well, fret not, the intercalated cells that are also found in the DCT, this is our second type. So if they're not principal cells, they're intercalated. These guys are going to focus a little bit more on acid-base balance. So they will either secrete more hydrogen ions into the tubular fluid, which will become the urine. So we will thereby lower our hydrogen ion concentration, or we can do the opposite. Maybe we are not breathing enough, or maybe we have vomited a lot. That's really the only conditions where people end up with alkalosis is whenever they vomit up all their stomach acid and then they're just left with the alkaline tide from the pancreas. So if that happens and we end up alkalotic, the other thing you can conversely do is secrete HCO3, the bicarbonate, to reverse that problem. So let's just take a look at this. Let's start kind of here. Right, so we'll start with a too much acid problem, right? So maybe the body's producing too much, maybe we're diabetic, maybe we are holding our breath and we have a breathing issue, I don't know. So that's going to increase hydrogen ion concentration, that's going to drop the pH, because remember those two things are inverse. And we're going to, of course, do our respiratory compensation, so we should breathe more if we're trying to get rid of acid and CO2. But the thing that is new to us, right, in this figure is the renal compensation. What are the kidneys going to do? Okay, so we are going to secrete hydrogen ions and try to save bicarbonate ions. Okay, so basically, if we look at our kidney here, we can see that, hey, hydrogen ions are being secreted, so that's going to help bring our pH back up to normal. Okay, so yay, pH is back to normal. Homeostasis is restored. I hesitate to even go over the other part of this if we end up alkalosis again, because most of your patients, if you plan on working clinically, most people are going to come in with acidosis, with diabetes on the rise and the obesity epidemic in the country. More of your patients are going to be acidotic than alkalotic, but people can come in with an alkalosis, meaning that their pH is too high, they have too much bicarbonate, right, and not enough hydrogen. So again, vomiting or some, get hit in the head and you have a brain breathing disorder because you've messed with the, Pontine center or something like that. But again, probably not as many of your patients are going to have this problem. Elevated bicarbonate, so pH is too high. The respiratory compensation would be breathe less. And then again, the renal compensation would be to preserve the hydrogen ions and to secrete the bicarbonate. And that's what we see happening here in the kidney. So instead of secreting the hydrogen, we're secreting the bicarbonate. And that is going to decrease our pH and bring us back to normal. And then yay, homeostasis is restored. Again, this is brought to you by the carbonic acid equation, so please do not forget that thing. And again, now we know that it's not just the job of the lungs to correct acid-base balance. The kidneys will help us out when the lungs cannot. Okay, one more mechanism. So we are on to antidiuretic hormone. The other name for antidiuretic hormone is also arginase vasopressin. It is a powerful vasoconstrictor. It also is powerful in promoting water reabsorption. This is one of your two posterior pituitary hormones. So ADH, the other one is oxytocin, and you're in for a treat because we're going to talk about that one next in the reproductive system. So we finally come around full circle, and we are talking about our posterior pituitary hormones. So antidiuretic hormone is very important in fluid balance, and its target tissues are going to be the distal convoluted tubule and the collecting duct. This is why I started the lecture with a review of our medullary osmotic gradient. So again, we can see in both figures here, we've got two side by side. Again, going from cortex to medulla, we can see the scale is the same on both. Again, at the top of the collecting duct, just like at the top of the nephron loop, the concentration of the interstitial fluid in the kidney is 300 milliosmoles. That's our normal. But as the collecting duct passes through the renal medulla, as the collecting duct continues to pass through the renal medulla, it's going to be subjected to that same gradient. So this is going to give us one last chance to reabsorb any additional water. Okay, so the mechanism by which this works is antidiuretic hormone that's released by those neurohormones in the hypothalamus that travel down to the posterior pituitary, where they're actually allowed to get incorporated into the blood. We're carried down to the renal tubules. And what's going to happen there is that ADH is going to act on our collecting ducts. Okay, and they're going to insert aquaporins. So we were familiar with aquaporins. They were transporting water. We saw these in the PCT. We saw these in the descending thin segment of the loop of Henle. So the cells there have aquaporins at the ready. For the collecting duct, it may or may not have aquaporins. It just depends on the presence of antidiuretic hormone. So if there's antidiuretic hormone present, more aquaporins will get inserted into the collecting duct here, and we'll reabsorb more water. If we don't have any antidiuretic hormone that is secreted, then we get no aquaporins. So that's what this panel is showing. No aquaporins, no extra reabsorption. So it just depends on what you did that day and your body's needs. So again, we have some level of obligatory water loss. We have to dilute those nitrogen wastes in some quantity of water. So you have to pee every day. Okay, but depending on what you drank that day and your hydration status, that will dictate how much you pee. So if you are hydrated, you will pee more. If you're dehydrated, your antidiuretic hormone is going to be at work and trying to pull that water in.